Good morning, students. Today we are going to see regulation of respiration. See, regulation of respiration is done by three ways. One is neural regulation, other one is chemical regulation, other one is non-chemical regulation. So today we will see what is the neural regulation of respiration. So the neural regulation is done by two control mechanisms. One is voluntary control, other one is automatic. See, voluntary control usually it is evident in breath holding. When you hold the breath, you have a voluntary control. Or if you are asked to do hyperventilation, you can increase your inspiration and expiration fastly. Or in some yogas, you can do this voluntary control. Here the center is cerebral cortex, which is voluntary and it is under the will. And from the cerebral cortex through the tract corticospinal tract, they terminate in the spinal cord motor neurons. One in the cervical, that is phrenic motor neurons, that is going to supply the diaphragm, which is the muscle of inspiration, and another is the motor neurons in the thorax, so in the thoracic segments, and this is going to innervate the external and internal intercostal muscles. Okay, so this is a voluntary control mechanism. So from the cerebral cortex through the corticospinal tract, it is controlling the motor neurons of the respiration, phrenic motor neurons and thoracic motor neurons, which is going to innervate the muscles of respiration. This is voluntary control. And most of the time we have the automatic control mechanism. Okay, the automatic control mechanism, it is lying in the centers in our brainstem. So in our brainstem, so this is the pons and this is the medulla and this is the spinal cord. Okay, so in the medulla we have the respiratory neurons that are controlling the automatic respiration. We have two groups of neurons. One is the dorsal respiratory group neuron and another one is the ventral respiratory group neurons. On both the sides you have dorsal respiratory group neuron and ventral respiratory group neuron. This is DRG, which is dorsal respiratory group neuron, and it contains inspiratory neurons. So it is responsible for inspiration. And the ventral respiratory group neuron, it contains expiratory neurons and it is responsible for expiration. And now the impulses from the DRG will be supplying the motor neurons in the spinal cord. Either it is phrenic motor neurons in the cervical segment or it is the motor neurons in the thoracic segment. Okay, so these motor neurons, the phrenic motor neurons and the thoracic motor neurons, they are going to innervate the muscles of inspiration and also expiration. Okay, expiration is the motor neuron supplying the internal intercostal muscles. And the VRG is usually, which is responsible for expiration, it is inactive, and DRG is active. So as inspiration is an active process, expiration is a passive process. Expiration is just nothing but the elastic recoiling of the lungs. Now, a portion of the VRGs have been divided into three portions. One is rostral, medium and caudal. And the major function is to control the expiration. And in and around the DRG and VRG, we have a pacemaker neurons. And these pacemaker neurons are called as pre-Bolzinger complex. So the pre-Bolzinger complex generate the pattern, okay, the respiratory pattern, or generate the rhythm. So they are also called as CPG, that is central pattern generator. So this DRG and VRG ultimately project to the CPG, central pattern generator, and this also have a rhythmic input to the phrenic motor neurons. Okay, phrenic motor neurons, that is controlling the inspiration. So the CPG or the pre-Bolzinger complex, it is also called the central pattern generator. It is containing the pacemaker neurons. So the pacemaker neurons for the respiration, they generate the rhythm or initiate the rhythm of respiration is pre-Bolzinger complex. Now these respiratory centers are in turn modulated by the Pontine centers. So these are the Pontine centers. So here you have the two Pontine centers. One is the pneumotaxic center, other one is the apneustic center. So this one, in the upper pons, you have the pneumotaxic center, and this one is the apneustic center. So normally the pneumotaxic center has an inhibitory output to the apneustic center. So what is the function of apneustic center? Apneustic center constantly inhibit the DRG. Okay, so constantly stimulate the DRG. So if DRG alone is stimulated, what will happen? There will be prolonged inspiration, which we call it as apneusis. That is prolonged inspiration. Inspiration alone is very active when it is stimulated. So this constant discharge is inhibited by the pneumotaxic center. So what is the function of pneumotaxic center? Pneumotaxic center limit inspiration. Limit inspiration, because we have to have a limit on inspiration so that the next process expiration will happen in a passive manner. Okay, so now if the pneumotaxic center is stimulated, it is going to inhibit the apneustic center, thereby the inspiration is limited, and that is the role of the Pontine centers. So Pontine centers, they modulate or they modify the inspiratory signals from the medulla to the spinal cord. That is the role of the Pontine centers. So we have Pontine centers. Here we have two Pontine centers, and we have the medullary centers. There we have two things, one is DRG, VRG and CPG. CPG is central pattern generator. Now what happens if you make a sectioning? Okay, so what is the change happening in the respiration when you make a section? And here we have to have an idea about the vagus nerve. What is the role of vagus in respiration? So vagus nerve normally it will inhibit the DRG. That is a function of vagus nerve. So what happens when there is overinflation of the lungs? So they cause a stretching of these stretch receptors, and these receptors normally in normal inflation also these receptors get stretched and they send signals to the vagus nerve. The vagus nerve will reach the DRG. DRG is actually, it is also called as nucleus tractus solitarius. So the DRG, dorsal respiratory group neuron, is present in nucleus tractus solitarius. It is a chief sensory nucleus where it is regulating many of the body functions, not only respiration, it is also the vomiting center, and so it is center for so many functions in our body. Now this DRG is inhibited by the vagus nerve. When the vagus will be stimulated, when there is inflation of the lung, this causes stretching of the airways, and in the airway we have a receptor called stretch receptor that will stimulate the vagus nerve, and the vagus nerve will inhibit the DRG. So what is the role of vagus? Vagus inhibit the NTS, okay, or DRG, dorsal respiratory group neuron. So the role of vagus is also like pneumotaxic center, which will limit inspiration. Now inspiration is limited. Now with this background we go for sectioning and we will see what are the changes taking place in the respiratory signal. Now if you make a section above the pons, all the centers are intact, so you will have a normal respiratory signal. And if you are making a section in between the pons, that is between the pneumotaxic center and apneustic center, what happens? There is no control over apneus, so inspiration will be lengthier. Okay. Now you are cutting the vagus nerve also. In the same section, you are cutting the vagus nerve. Vagus nerve is also which will limit the inspiration. So what will happen without vagus? The inspiration will be lengthier and it will be persistent for some time. Okay. This type of breathing is recorded when vagus nerve is also cut. So this is with vagus, and section is between the two centers in the mid pons between the pneumotaxic and apneustic center. And along with that section, if you are cutting the vagus, then the inspiration will be very lengthier. So this is due to the strong activation of apneustic center, and also there is no vagus to limit the inspiration. Now if you are making a section in the in between the pons and the medulla, between the pons and the medulla, if you are making a section, what will happen to the respiratory signals? It will be there, but it will be irregular. So the depth and rate of respiration will get altered. And if you are sectioning below the medulla, this is a sectioning below the medulla, what will happen? There is no respiration. So this section 1 is above the pons, section 2 is mid pons. In section 2 we have two recording, one is with vagus, other one is without vagus, and section 3 is between the pons and the medulla, this section. And here you have a irregular breathing pattern, and section 4 is below the medulla where there is no respiration. So these are the effects of sectioning at various levels, and they are studied, and based on this we are able to assign the function of each. So here the chief respiratory center is located in the medulla. So in the medulla you have dorsal respiratory group neuron for inspiration, ventral respiratory group for expiration, and they do project into the CPG, that is central pattern generator, which is containing a pacemaker, which is one controlling the rhythm of respiration. And you have to remember the two neurons. One the neuron, the cervical segment, which is the phrenic motor neuron, which is giving rise to phrenic nerve supplying the diaphragm. The other neuron is the motor neuron present in the thoracic segment that is supplying the intercostal muscles. Okay, so tomorrow we will see the chemical regulation. Okay, so this is what we have seen today is the neural regulation.